Hello everyone, welcome to our YouTube platform Concept and Fact of Life Sciences. Today in this video we are going to discuss about the protein structure, that is primary, secondary, tertiary and quaternary structure. We will also discuss about the Ramachandran plot and the position of protein secondary structures on the Ramachandran plots. We will also discuss about the chaperones and chaperonins. Let's start with the basics, that is the protein primary structure. Now the primary structure of protein, it is stabilized by the peptide bonds. And these peptide bonds, these are the type of covalent bond. How these peptide bonds are formed? We are taking an example. We are having two amino acids adjacent to each other. This is having a basic framework structure as well as the R chain, and depending upon this R chain, we are having different types of standard and non-standard amino acids. So the carboxylic end of one amino acid, it is in front of the amino group of the another amino acid. There is a condensation reaction that lead to the removal of H2O, OH from this. There is a removal of one H from this. There is a formation of a covalent bond that is called as the peptide bond. So we are having the formation of peptide bond over here. This is representing a peptide bond. So as we all know that the protein, it is a polymer of amino acids. So the monomeric units, the monomer of protein, it is the amino acid. So the amino acid, they bound by the peptide bond that leads to the formation of a polypeptide or proteins. One end is always NH2 amino end and the other end is always carboxylic end. So this is representing a polypeptide. Now if we take an example, or if we take a amino acid from this polypeptide chain, suppose we are taking this amino acid. Now this bond, it is the peptide bond, which is known as the omega bond. Well, this is another peptide bond which is known as the omega bond. Now the rotation of polypeptide, it is not possible around the peptide or the omega bond. We can state over here that the rotation is not possible around the peptide bond. The peptide bond, still they are the single bond, but still they are not allowing the rotation. Why? Because these peptide bonds, they are the planar structure having the partial double bond characters. They are having partial double bond characters which do not allow the rotation. So due to the partial double bond characters, the rotation is not possible around this peptide bond or this peptide bond or this peptide bond. So the rotation is not possible around the peptide bond, but still the protein, they readily adopt the secondary structure and the tertiary structure. So the rotation should be there. So the rotation, if it is not possible around the peptide bond, but the rotation is possible around some other bonds, like between the N and C. The C, which is adjacent to the functional group, it is called as the C alpha. तो ये alpha carbon है. The rotation is possible around the N and C alpha, and this rotation, it is called as the phi rotation. Another rotation which is possible is between the C alpha and C prime, and this rotation, it is known as the psi rotation. So the rotation is not possible around the peptide bond due to the partial double bond characters, but the rotation is possible around the N-C alpha, that is called as the phi bond, and the C alpha and C prime, that is called as the psi bond. So the rotation is possible around the N and C alpha, that is called as the phi rotation, and C alpha and C prime, that is called as the psi rotation. These rotations, they were firstly discovered by scientist G.N. Ramachandran. According to scientist G.N. Ramachandran, the secondary structure of protein, it depends upon the protein primary structure, and how much this rotation will occur, that is the phi and psi, it depends upon the R chain of amino acids. इसके बारे में अपन आगे डिस्कस करेंगे जब अपन रामचंद्रन प्लॉट्स के बारे में बात करेंगे. सो दीज रोटेशंस दे वर फर्स्टली डिस्कवर्ड बाय द साइंटिस्ट Ramachandran that allows the protein to adopt the secondary and the tertiary structures. So if we talk about the secondary structure of protein, we are having two important type of structure, that is called as alpha helices and the beta sheet. जैसे आपने देखा कि प्रोटीन की जो प्राइमरी स्ट्रक्चर है, they will get stabilized by the covalent bond, that is called as the peptide bond, while these secondary structure they get stabilized by the another type of bonds, that is the non-covalent bond known as the H bond. So the secondary structure of protein gets stabilized by the hydrogen bonds. These alpha helical structure, they remain abundantly present within the globular protein like alpha keratin, G-actin, which plays an important role in the cytoskeleton, as well as in the hemoglobin. These alpha helical structures, they were firstly discovered in the alpha keratin. That's why they are named as alpha helices. If we talk about the structure and the bonding of these alpha helices, this structure again it is representing a polypeptide or the protein chain. Within this polypeptide, if we are having an amino acid, it is suppose an amino acid. One amino acid in the alpha helical structure can form two hydrogen bond. It can form hydrogen bond with N plus 4 amino acid, like it is the N plus 1, 2 and 3 we are leaving, and it will form the hydrogen bond with N plus 4, and it can also form the hydrogen bond with N minus 4 amino. So one amino acid in alpha helical structure can form two hydrogen bond, one with N plus 4, other with N minus 4 hydrogen bond. Simply, as we already discussed in our previous lecture, that they are generally formed between the two electronegative atom in which there is a sharing of hydrogen atom. So if here the electronegativity of the oxygen is more, so the C will act as a hydrogen donor, it will act as a hydrogen acceptor. Similarly, this nth amino acid, it will also act as a hydrogen donor for N minus 4 amino acid. So one amino acid in the alpha helical structure, they forms two hydrogen bonds. Now these alpha helical structure, they are of two types. There is right-handed alpha helical structure as well as the left-handed alpha helical structure. These helical structure, they are formed when all groups, they will rotate in a similar direction. If the rotation is in the clockwise direction, then it will lead to the formation of right-handed alpha helical structures. Well, if the rotation it is in the counterclockwise direction or in the anti-clockwise direction, then they will lead to the formation of left-handed alpha helical structure. Right-handed alpha helical structure, they are more stable. Now, these alpha helices, they are also known as 313 helices. Why? Because the two amino acids involved in the formation of hydrogen bonds, they are 3 amino acids, that is 1, 2 and 3. They are 3 amino acids and 13 atoms away from each other. So this 3, it is representing the amino acid, and 13 atom means we are having 1, 2, 3, 4, 5, 6, 7, 8, 9. We are taking the NH of this amino acid and carboxylic end of this amino acid. So there is 12 and 13. So the two amino acids which are involved in the formation of hydrogen bond, they are 3 amino acid and 13 atoms away from each other in the alpha helix structures. That's why they are also known as the 313 helices. If we talk about the structure of alpha helices, this diagram, it is representing the helix, alpha helices. In one helix, there is 3.6 residues of amino acid, 3 complete amino acid. The distance between the two amino acid involved in the hydrogen bond is 3, and 0.6 partially of nth and partially of n plus 4 or n minus 4. So in one helix, there is 3.6 residue of amino acid. The number of amino acid in one helix, it is 3.6, as we already stated. The pitch of the helix, that is the helix height, it is 5.4 angstrom. There is a formation of hydrogen bond, as we already stated that there is a formation of hydrogen bond between the n and n plus 4 and n and n minus 4. So the helix height is 5.4 angstrom. So what will be the distance between the two amino acid in the alpha helical structure? We can easily find out that 5.4, it will be divided by the amino acid residue in one helix. It will come around 1.5 angstrom. So the distance between the two amino acid in the alpha helical structure, it is 1.5 angstroms. Now another type of secondary structure, it is the beta sheets. These are also known as the beta plated sheets. Why they occur in the plated form? Because if they will not occur in the plated form, the R chain of the amino acid will not allow the formation of hydrogen bonds. So they will also known as the beta plated sheets. Now we are having two type of beta sheets. One are known as the parallel beta sheets, while other is known as the anti-parallel beta sheets. Parallel means if we are having the carboxylic terminus towards one end and the NH2 terminus towards other end, then they are known as the parallel beta sheet. And if we are having one carboxylic, one NH2 terminus towards one end, while other NH2 and COX carboxylic terminus towards the other end, then it is known as the anti-parallel type of beta sheet. So we are having parallel and the anti-parallel beta sheets. Now the anti-parallel beta sheets, they are more stable as compared to the parallel beta sheets. Why? Why the anti-parallel beta sheets are more stable? By looking into the structure, we can easily find out. जैसा कि अपन ने पहले अपने लेक्चर्स में बात की थी, जब अपन ने नॉन कोवेलेंट बॉन्ड में हाइड्रोजन बॉन्ड्स को पढ़ा था, बॉन्ड एनर्जी हाईएस्ट होगी कब हाइड्रोजन बॉन्ड की? जब H डोनर, H एक्सेप्टर और H एटम तीनों एक ही प्लेन में होंगे, बिकॉज द हाइड्रोजन बॉन्ड्स दे आर द डायरेक्शनल बॉन्डिंग. वी कैन से इफ द H donor, H atom and the H acceptor, if they are in a same plane, the bond is strongest and the bond energy is highest. So we can say in case of the anti-parallel sheet, there is a planarity. There is a planarity in the hydrogen bonds, which forms more stable structure. While if we talk about the parallel beta sheets, the all three ones, that is the hydrogen donor, acceptor and H atom, they are not in the same plane. So the bond energy is slightly lesser as compared to the anti-parallel beta sheets. So they are less stable structure. So why the anti-parallel beta sheets are more stronger? Because they have planarity in their hydrogen bonds. Now these beta sheet structures, they are found in case of beta keratin, antibodies, immunoglobulins, as well as in case of the porine proteins. We are having parallel and anti-parallel beta sheets, जो अपन ने पहले ही डिस्कस कर लिया. The distance between the two amino acid in beta sheet structure, it is 3.5. Alpha helices में अपन ने बात की, the distance is 1.5 angstrom. While in case of the beta sheet, the distance between the two amino acid, it is 3.5 angstrom. ये याद रखना है अपने को. Anti-parallel beta sheets, they are more stronger due to the planarity of hydrogen bonds. So this is all about the beta sheet structures. Next structure, it is called as the beta turns, which is also known as the beta bands or the hairpin turns. Now these beta turns or hairpin turns, they played a vital role in the formation of globular structure. These turns, they generally found in the surface, on the surface of the globular protein, where they connect the two anti-parallel beta sheets. There is a connection of two anti-parallel beta sheets through the beta turn. In case of the beta turn, here the nth amino acid, it will forms the hydrogen bond with N plus 3. Common amino acid which remain present in case of the beta turns. Onto the second position, we are having proline, while onto the third position we are having glycine. जब अपन अमाइनो एसिड्स की बात की थी तो अपन ने कहा था glycine and proline amino acid, they are also called as the alpha helix breaker because they do not fit in the regular structures. तो यहाँ पे जो proline है ये turns को induce करता है, while the glycine it provides the flexibility. जो C1 है it will form the hydrogen bond with the C3, we can say first number जो amino acid है nth amino acid, it will form the hydrogen bond with N plus 4, we can say N plus 4, and N plus 2 and 3 it will not remain allowed the formation of hydrogen bonds. Commonly we can say it is made up of 4 amino acids. So this is all about the beta turns or the reverse turns. Now if we talk about the Ramachandran plot, this is most important part of this lecture. These plots, these were firstly discovered by scientist G. N. Ramachandran. That's why they are also known as Ramachandran plots. अपने इनिशियली बात की थी कि जो पेप्टाइड बॉन्ड है that do not allows the rotation due to the partial double bond character, but the rotation it is possible around the N and C alpha carbon. That rotation is called as the phi rotation, and the rotation is possible around the C alpha and C prime carbon. That rotation is called as the psi rotation. अब ये कितना rotation होगा, it depends upon the we can say R chain of amino acid. So y axis पे अपन ने क्या किया, psi rotation ले लिया, x axis पे अपन ने phi rotation को ले लिया. अब rotation कितना हो सकता है, it can occur 180 degree plus to minus 180 to plus 180 in case of phi. So it can occur from plus 180 to minus 180. इतना रोटेशन हो सकता है. अब कितना रोटेशन होगा, इट डिपेंड्स अपॉन द आर चेन ऑफ द अमाइनो एसिड. यही साइंटिस्ट रामाचंद्रन ने कहा था कि द सेकेंडरी स्ट्रक्चर ऑफ द प्रोटीन इट डिपेंड्स अपॉन द प्रोटीन प्राइमरी स्ट्रक्चर्स. So in this plot we are taking the psi rotation on y and phi rotation on x axis. We have divided this plot into the 4 parts. We are having some disallowed or non-allowed regions, while we also having some allowed region depending upon the values of psi and the phi. psi रोटेशन कितना हो सकता है, प्लस 180 से माइनस 180. इसी तरह से phi रोटेशन भी 180 से माइनस 180 तक हो सकता है. पर कुछ रीजंस हैं जो डिसअलाउड हैं, इस डायरेक्शन में जो रोटेशन है वो पॉसिबल नहीं है. दैट इज ड्यू टू द आर्चिन और स्टेरिक हिंड्रेंस, वी कैन से. The least steric hindrance it is imposed by the glycine. Why? Because the glycine it is only having the H into its side chain. So R value is equals to H. तो R chain इसकी minimum H है, H है, that do not impose any steric hindrance. So it can occur any quadrant of this Ramachandran plot. While the least occurrence is of the amino acid proline, because the proline it is also known as the alpha amino acid. अब इसको अल्फा इमिनो एसिड क्यों कह रहे हैं, बात करेंगे इसके बारे में. होता क्या है, या अपन कह भी चुके हैं, बता भी चुके हैं आपको इसके बारे में. होता क्या है, यहाँ पे एक रिंग स्ट्रक्चर का फॉर्मेशन हो जाता है विद इन द अमाइनो एसिड. That's why it is called as the alpha amino acid, and due to the formation of this ring it impose maximum steric hindrance that do not allow much psi and phi rotations. So it occupies least space in this on this Ramachandran plot. Maximum space is occupied by the amino acid glycine. So if we talk about the protein secondary structure onto this plot, we are having anti-parallel beta sheets over here, parallel beta sheets over here. We are having polyprolines over here. So if we talk about this quadrant, here we are having the positive value of psi. We are having psi, that is on the y-axis we are having the positive value of psi, while there is a negative value of phi. So on this particular condition, or on this rotation, we are getting anti-parallel beta sheet, parallel beta sheet, and the polyproline structure. Well, if we talk about this part, it is having the psi value that is in the negative way, and phi is also negative. Both are negative. Under these conditions, you are getting the right-handed alpha helical structure, and also we are having the 310 helices there that are between the right-handed alpha helices and the beta sheets. If we talk about this quadrant, it is of the left-handed alpha helical structure. Here we are having the positive value of phi as well as the positive value of psi. So both of the value they are positive, and under this condition we are getting left-handed helical structure, and this is the most disallowed region, which is having the positive value of phi and the negative value of psi. This is the most disallowed regions. So this is all about the Ramachandran plot. अब क्वेश्चंस इसके ऊपर आपसे सीधा पूछा जा सकता है कि बीटा शीट कौन सा क्वाड्रेंट ऑक्यूपाई करते हैं, अल्फा हेलिक्सेस कहाँ पे होते हैं, या आपको प्लॉट दे दिया गया और मार्क करके पूछ लिया गया कि ये रीजन किसको शो कर रहा है. तो इस तरीके से क्वेश्चंस इसके ऊपर पूछे जा सकते हैं. सो दिस इज ऑल अबाउट द Ramachandran plots. Now we are also having the tertiary and the quaternary structure of the protein. The folding of secondary structure over themselves lead to the formation of tertiary and the quaternary structure. प्राइमरी स्ट्रक्चर स्टेबलाइज हो रहे थे कोवेलेंट बॉन्ड से, पेप्टाइड बॉन्ड से. सेकेंडरी स्ट्रक्चर स्टेबलाइज हो रहे थे हाइड्रोजन बॉन्ड, नॉन कोवेलेंट बॉन्ड से. While the tertiary and the quaternary structure, they are stabilized by the hydrophobic interaction. The major driving force is the hydrophobic interaction, while all other type of bonds like the H bonds, van der Waals bonds, as well as the sulfur bonds, sulfide bonds, they will provide the further stability. Now there is an interaction. These bonds, we can say these structures, they are generally formed by the interaction between the R chain. अभी तक जो interaction हो रहा था, that is between the backbone of the polypeptide. Now the interaction, it will occur between the R chain of amino acid. If the protein is made up of single subunit, then the final structure will be tertiary structure. For example, the myoglobin, which is the oxygen carrier in case of muscles. While if the protein is multi-subunit, means अगर एक से ज्यादा सबयूनिट का बना हुआ है, then the final structure will be the quaternary structure. We can take the example of hemoglobin over here, which is made up of 2 alpha and 2 beta subunit. So it is made up of more than one subunit. So the final structure will be the quaternary structure. So this is all about the tertiary and the quaternary structures. Another concept is of the chaperones and the chaperonins. These chaperones, they help in the protein folding, refolding, as well as in the assembly of protein. So we can say they are involved in the protein homeostasis, that is also called as the proteostasis. If we talk about the chaperones, these chaperones, they generally bound to the newly synthesized, suppose it is a mRNA having the ribosome, there is a synthesization of a polypeptide. These chaperones, they generally binds to these newly synthesized polypeptide and do not allows the folding of protein until the complete synthesization of polypeptide. So they generally assist in the protein folding, we can say, and the example is the HSP70, heat shock protein 70 class of proteins. While other category, it is of the chaperonins. Now these chaperonins, they are the type of chaperones that generally forms a barrel like structure through which a misfolded protein or non-folded protein can pass through, and these barrels, they provides the adequate environment for the protein folding. So we can say these chaperones, they are directly involved in the protein folding. They provides the adequate environment for the proper protein foldings. They generally forms a cylindrical like structures that allows the protein to fold in a proper way. If we talk about the example, we are having the grow EL and grow ES, which is found in case of the prokaryotic system as well as in case of the eukaryotic mitochondria, because the eukaryotic mitochondria, it is also of the prokaryotic in origin, जो आगे अपन बात करेंगे. While in eukaryotic cytosol, we are having TRIC CCT system, which is a type of chaperone. So this is all about the protein structures, Ramachandran plots and chaperones. Thank you very much.